Continuing with the digestive system, we've just left the stomach. So the next thing we're going to look at is the accessory organs whose secretions go into the first part of the small intestine. One of those is the pancreas. It's found a little below and behind the stomach. Looks sort of like a music note or a comma. We call the wide end the head, and that's nestled into a curve of the small intestine. And then it gets narrower, we call that the body, and tapers out into a tail. Its function is to secrete pancreatic juice, which is going to have several functions in digesting. And all the juice is secreted from the cells into ducts. They combine into one big pancreatic duct that enters the small intestine. Here's the head, body, and tail, pancreatic duct. It does actually split here. One branch goes directly to the small intestine. The other combines with the bile ducts. So if you need the pancreatic juice but not the bile, you can still get it. On the microscopic level, remember there's two different things going on in the pancreas. You've got your endocrine cells grouped in islets that make your insulin, glucagon, and so on. And then your acinar cells. Each little cluster is called an acinus. That's these. They secrete into that little space, and their secretions continue out into ducts. Pancreatic juice includes several digestive enzymes that we'll take a look at, and some buffers to neutralize stomach acid. Pepsin needs to be at a low pH to work, but the enzymes secreted by the pancreas work better at a neutral pH, plus the acid could damage the small intestine. It doesn't have the same protection as the stomach. Too much mucus would make it harder for substances to get absorbed. The pancreatic enzymes correspond with the four major categories of organic molecules: carbs, lipids, proteins, nucleic acids. You've got amylase for carbs, same as the salivary amylase. It breaks polysaccharides into disaccharides. Lipase digests triglycerides. It cuts the fatty acids off them. That makes them all 1/3 the size, and that's small enough to absorb. Nuclease breaks nucleic acids into nucleotides. They're always going to be in your food because a lot of food is made of cells. So it has a nucleus with chromosomes in it made of nucleic acids. And then there's a group that digests proteins. You'll see them sometimes called proteases as a group, or proteolytic enzymes. There are several different proteolytic enzymes. These are in addition to the pepsin from the stomach. I'm going to prepare this as an explanation of why we need multiple enzymes. The major ones are trypsin, chymotrypsin, and carboxypeptidase. If you remember, your enzymes usually end in ACE, but some of these digestive ones were the early discoveries of enzymes, and we didn't realize how many there were and that we needed a special ending for it. Now, carboxypeptidase goes up to a chain of amino acids and just lops off the last one. Now, proteins can be hundreds of amino acids long, so it could take it a while to just do that. What the others do is break the protein into smaller pieces. Each one recognizes certain amino acids, joins and breaks it there. So let's say trypsin recognizes the orange ones. That's already given us one free amino acid And now we have 3 pieces, so the carboxypeptidase could be working on, you could have 3 of it working on all these pieces at the same time. And then let's say chymotrypsin recognizes the blue ones. That gives us even smaller pieces.
So they all kind of work together to break it apart?
Yeah,
Okay.
they all go in different spots and snip it.
Okay.
Now, because breaking down protein that's part of a cell, like in a pancreatic duct, could be bad, like the pepsin, these are secreted in inactive forms. So pepsinogen becomes pepsin, there's a trypsinogen that becomes trypsin. They're activated once they enter the small intestine. So, any other questions on pancreatic enzymes?
They are all activated whenever they go into the small intestine?
The protein ones, the amylase and lipase, we don't have to worry about because the things they break down aren't generally on the surface of cells, the polysaccharides and triglycerides. Next is the liver. It's divided into 4 lobes, the largest of which is the right lobe. Right and left are the only ones you can see from the front. Now, the liver is actually kind of triangular in cross-section, where it's got a top, a front, and then it's kind of got a back bottom that's one side. If we flip it up, so you're looking at that back/bottom side, we call it postero-inferior, put together 2 of those terms. Then we can see the other 2 lobes. So here's the top of the liver now. So we still have left over here, right over here. There are 2 landmarks along the back of the liver. The inferior vena cava, and the gallbladder. The quadrate lobe is rectangular. It's near the gallbladder. It's a quad for 4 sides. The caudate lobe, so you still have the ligament separating left over here. That means tail, because it has kind of this little hook that comes around the inferior vena cava. In between the 2 is an area called the porta hepatis, which is basically a hilum. And I've seen some textbooks call it the hilum rather than this fancy term that means liver door, basically. This is where tubes connect. Those are the hepatic artery, which is a branch from the celiac artery. That's bringing oxygenated blood into the liver. The hepatic portal vein, that's bringing the blood from the digestive tract. So it's low oxygen because it's already delivered the oxygen to the alimentary canal, but it's got the nutrients in it. So those are both flowing in. Then there's also tubes for the bile to flow out. The first part of that is the common hepatic duct. called common because there's a duct coming from the left lobe and one coming from the right lobe, and this is where they join to make one duct. That's the only thing that's coming out of the liver at the porta hepatis. The blood coming out would be in the hepatic veins going into the inferior vena cava. So here's the gross anatomy of the liver. Front view, where we can see ligaments separating right and left lobes. The gallbladder peeks through in the front. There's the inferior vena cava. Then when we flip it up, we bent the inferior vena cava. So this is the part that would be going down. Here's the caudate lobe. Here's the quadrate lobe. So you've got still that ligament separating the left lobe. So caudate and quadrate are parts of the right lobe. But naming them allows us to be more specific about where something is. And so then here's the hepatic artery, hepatic portal vein, and common hepatic duct up here. You can actually see the right and left branches. And then just this part of it is the hepatic duct. When the gallbladder splits off, we get different names. So any questions on the gross anatomy of the liver?
Like, what's the outline of the caudate lobe?
It's from the porta hepatis, so these are kind of sticking out a little. There's some space underneath them. So it follows along the gallbladder's duct behind there, down the ligaments, and then this little hook around the inferior vena cava, and then to about here. On the microscopic level, liver tissue is organized in lobules, microscopic repeating structures. In some species, these are neatly outlined in connective tissue and make obvious hexagons. So they're often drawn that way in books. The pig is the animal where we studied the liver most closely. The human lobules aren't quite as obvious, but this is a good way to understand them. So they're made up of hepatocytes, the cells that carry out the liver's functions. The other cells within them are Kupffer cells, which are macrophages that stay in the liver. Hepatocytes would be arranged kind of in radiating lines. They're cuboidal. Something like that. What's in between is blood flow and the bile flow. So bile is the digestive secretion of the liver. The hepatocytes produce it, and there's a whole bunch of little tiny gaps between them called bile canaliculi that can collect it. That goes to, it should say bile ductules, little ducts, at the corners of the lobules. The bile ductules join together to make the hepatic duct. Now the blood flow corners of lobules will also have branches of the hepatic artery and portal vein. And I'll just do these in blue for simplicity. Capillaries that come out join together run in between rows of hepatocytes. Let's say the vessels over here too. Those spaces are called sinusoids. Like we saw in the cardiovascular chapter, large, irregularly shaped and more permeable capillaries. All the sinusoids flow toward the middle of the lobule into a central vein. And then the central veins are all going to come back together to form the hepatic veins and go into the inferior vena cava. So blood from most of the digestive tract goes into the liver, out into